Hello and welcome to another Tissue Gnostics webinar. My name is Carly Briffa and today we're joined by our guest Dr. Amirreza Mahbod from the Research Center for Medical Image Analysis and Artificial Intelligence at the Danube Private University to present his recent collaborative research on deep learning for improved nuclei segmentation in microscopic images. Thanks for joining us today, Amirreza.
Hello everyone. In today's presentation, I'm going to talk about our recent collaborative project entitled Deep Learning for Improved Nuclei Segmentation in Microscopic Images. This project was funded by FFG and it was a collaborative project between the Medical University of Vienna and Tissue Gnostic. This project was started in 2020 and ended in 2022. Before I talk about the project, I would like to introduce the main team members that were working in this project. As you can see in this slide, we had different collaborators who were involved in this project, but the core team members were Professor Isabella Eilenger from the Institute of Pathophysiology and Allergy Research at the Medical University of Vienna, myself, Amirreza Mahbod, where I did my postdoc with the frame of this project, Professor Georg Dofner from the section of Artificial Intelligence at the Medical University of Vienna, and Dr. Rupert Ecker from Tissue Gnostic. With introducing the team member, now we can talk about the project itself. But before going through the technical aspect of the project, I would like to give you some background information about histopathological images and how we can analyze them. The generic workflow of creating and analyzing histopathological images is shown in this slide. As you can see from the flowchart, we can extract different tissues and samples from different organs of the body. And then based on different staining and fixation protocols, we can prepare them and put them on glass slides. Then medical experts and pathologists can look at these glass slides with microscope, and then based on their knowledge and experience, they can analyze the images. But as you might expect from this manual process, it could be very time consuming. Also, quantification and statistical analysis with this approach would be very difficult, especially because of the large size of histopathological images. This method can also suffer from intra and inter-observer variability. To solve these issues, we can use automatic computerized approaches. And nuclei segmentation and classification plays an important role in computerized automatic histopathological image analysis. When we are talking about nuclei segmentation and classification, first we need to know the exact definition. Here we can see one input image, which is an image patch which is extracted from a whole slide image on the left side of this slide. Then we might need to separate the background and foreground just to identify the nuclei, as you can see in the middle part of this figure. If we are just interested to separate the foreground and background and we don't care about different objects in the images, then we are dealing with the semantic segmentation task. But in our case, because we are usually dealing with overlapping and touching objects, we also want to separate these objects or nuclei and label them with different numbers. And this is the main task of this project, and this is the definition of the instance segmentation. When we create this instance segmentation mask, we can use them for further and subsequent analysis. For example, the nuclei density or the number of nuclei or their shapes can be used for different medical and research applications such as breast cancer grading. To perform nuclei instance segmentation with computerized method, there are various approaches that have been proposed in the literature. They are ranging from classical image processing techniques to advanced machine learning and deep learning based approaches. But in order to summarize all of them, we can put all of these approaches in this flowchart, as you can see on the right. So as you can see, usually we have some data sets which are some histopathological images, and we want to perform nuclei instance segmentation or classification in this dataset. Based on the dataset, we might need to apply some pre-processing steps to make them ready to be sent to our classifier or segmenter. Then we have different approaches as the main workflow or algorithm to perform nuclei instance segmentation and classification, which as I mentioned earlier, they could be based on classical image processing techniques, machine learning approaches, or deep learning based approaches. And finally, when we have the results from these approaches, we might need to apply some post-processing to have the final instance segmentation results. In order to quantify the results and evaluate the performance, we can use well-defined evaluation indexes to evaluate the performance of our approach. In our project, we developed several algorithms to perform nuclei instance segmentation and classification. You can see the title and respective publications of these approaches on this slide. Because of lack of time, I just focus on the last one, which is our most recent developed algorithm to perform nuclei instance segmentation and classification, which also delivered the best performance among our approaches. The title is a dual decoder unit-based model for nuclei instance segmentation in hematoxylin and eosin histological images. I'm going to use the same flowchart that I introduced in previous slides to go through the workflow and show you the different steps that we developed in this specific project. I start with the dataset. We use 4 different datasets in this study. We use the cryonosec dataset, NewInsect dataset, monosec dataset, and monosac dataset. All these datasets are publicly available and can be used to develop algorithms for nuclei instance segmentation or nuclei instance segmentation and classification task. Among this dataset, we use the monosec dataset in order to pre-train our model, but we use the other dataset to further train, fine-tune, and evaluate the performance of our model. The statistics of these datasets are shown in the table on these slides. Here on this slide, we can see some example of different image patches on these different datasets, such as cryonosec, NewInsect, the monosec. We can see the original raw input images on the top, and we can see the respective nuclei instance segmentation or nuclei instance segmentation and classification on the bottom of this figure. The next step is pre-processing. We applied these 3 different pre-processing steps in this study. First, we normalize the intensity values and bring them to the common range between 0 and 1 to train our model. We also apply different augmentation techniques to artificially increase our training size. We use different color-based and morphological-based augmentation techniques in this study. We also created binary and distance map from the provided ground truth segmentation mask. We use this binary and distance map later on to train our model. You can also see some of these binary masks and distance maps in this slide. Then we have the main method that we developed in this study to perform nuclei instance segmentation and classification. The generic workflow of this approach is shown in this slide. As you can see, our method has 2 main parts, which are shown with 2 different colors, the blue part and the green part. The blue part is there just to perform nuclei instance segmentation. But if we want to perform nuclei classification on top of nuclei instance segmentation, we can also add this green part to the flowchart. So we go through different steps that we have in this flowchart. The main workflow to perform nuclei instance segmentation is shown in the top part of this figure. As you can see, in order to perform nuclei instance segmentation, we developed a dual decoder unit-based model which contains a single encoder and 2 different decoders. The encoder and decoders consist of convolutional layers, dropout layers, max pooling layers, and also transposed convolutional layers. We use the results from the output of these 2 decoders to form our nuclei instance segmentation masks. If we want to apply nuclei classification on the top of nuclei instance segmentation results, we can also use or add another encoder-decoder unit-based model to the workflow to perform classification on top of the results of the nuclei instance segmentation. Then we have the post-processing as the next step of the flowchart, and we apply two different post-processing based on the task. If we deal with the nuclei instance segmentation task, we just apply two simple post-processing, namely removing tiny objects and filling the holes based on morphological operations. And if we deal with the nuclei instance segmentation and classification tasks, beside those two post-processing steps, which I mean removing tiny objects and filling the holes, we also try to boost the segmentation performance with different ensembling approaches such as 5-fold cross-validation ensemble or test time augmentation. And also for one of the specific experiments, we removed the ambiguous areas from the created or final nuclei instance segmentation and classification task, and these ambiguous areas actually were provided in the data set itself. And finally, when we had our results, we needed to evaluate the results. To evaluate the performance of nuclei instance segmentation, we use well-known DICE score, aggregate Jacquard index, and panoptic quality scores. And to evaluate the performance of nuclei instance segmentation and classification, we also calculated the average panoptic quality score per class. And here are the experimental setup in this specific study. So we used the cryonosec, nunsec, and monosec challenge dataset to train and evaluate our model. Here are the results from the first experiments based on the cryonosec dataset. As you can see, our results in the last row of the table is superior based on all three evaluation indexes in comparison to the other state-of-the-art approaches. We can also see some visual examples of the ground truths and the predicted instance segmentation mask from our approach, just to visually inspect our results. In these slides, we can see the results from the next experiments based on the new insect dataset. And here we can see again that our method delivers superior performance based on DICE score, aggregate Jacquard index, and panoptic quality in comparison to the other approaches. We can again see some visual examples and comparison between the ground truths and the predictions which are derived from our approach. And finally, we can see the results based on the monosec experiment. And here you can see that we report the results based on the panoptic quality per class, per nuclei class. In this data set, we had 4 different nuclei classes, namely epithelial class, lymphocytes, macrophages, and neutrophil. And if you compare our results with the other state-of-the-art algorithm, you can see that our method is able to deliver very competitive results. And in particular, we were able to achieve the first rank in the post-challenge leaderboards of the monosec challenge, which shows the good performance of our algorithm, not only for nuclei instance segmentation, but also for nuclei instance segmentation and classification tasks. Besides developing algorithm for nuclei instance segmentation and classification, we also published a couple of publicly available datasets with the frame of our FFG project. You can see the name of these datasets and their statistics in these slides. I put the links for each individual dataset, so if you are interested to see more details about this dataset, please feel free to check them. You can, of course, use them for your research purposes and develop further and more powerful deep learning based approaches to perform nuclei instance segmentation or classification based on these datasets. We were also able to publish our results in well-known journal papers, as you can see in these slides. Just to show you some statistics of these publications, they had the accumulative impact factor over 46, and they have been cited for more than 650 times, which shows the good impact of our publication and of our work for the research community. Besides these journal papers, we were also able to publish 5 Congress papers. As you can see their names on these slides. In the frame of this project, we also participated in the number of medical image analysis challenges, mainly for performing segmentation and classification in various types of images, as you can see on these slides. And as you can see, in most of them, we were able to achieve very good ranking, which shows the good performance of our development methods for different tasks of classification and segmentation in medical images. With this slide, I would like to finish my presentation, and I would like to thank all the collaborators from different universities and institutes that collaborated in our project. I would also like to especially thank Austrian Research Promotion Agency to support and fund our project. Finally, I would like also to thank you for your attention, and if you are interested in this topic, or if you have any question about different projects or slides that I presented in this presentation, please do not hesitate to contact me. I provided my email address and also my LinkedIn profile, so I would be very happy to discuss with you further. Thank you and bye.